So now we're going to talk about what's called excitation-contraction coupling. That is the story that applies to skeletal muscle. So basically, the term coupling, when two events are coupled, it means that one event cannot happen without another. If muscle is excited, it will contract. And contraction can't happen without excitation. Does that make sense? Like just fancy way to express a pretty simple concept. So excitation happens at what is called neuromuscular junction, and we're going to try to draw it. So here, remember, skeletal muscle can only be excited by a nerve impulse. So this is the presynaptic terminal of a lower motor neuron. This presynaptic terminal forms, obviously, a synapse. Okay, so this is sarcolemma. And this kind of point of contact, and let me just draw it a little bit farther because I will need some structures here. Okay. So this point of contact, that synapse between the motor neuron and a muscle fiber, because this is the cell. I want you to understand what you see here at the bottom is the longitudinal section of a muscle cell. Like, I don't draw the bottom, but imagine the bottom is at the floor level, like the other side. It's like cylindrical and stuff, but you're looking at the cross section. Okay, 2D projection. So the structure here is called neuromuscular junction. Neuromuscular junctions can be found only in the skeletal muscle, period. Okay? So what happens? Well, first of all, of course, action potential will arrive onto the presynaptic terminal. Old story, you know that story. You know, calcium channels are going to open, and the influx of calcium will result in the release of, lo and behold, neurotransmitter. So this neurotransmitter that is released is always, without an exception, acetylcholine. Acetylcholine will open channels on the sarcolemma, and these are sodium, potassium. Now, since they are opened by a chemical, that's going to be ligand-gated channels, right? So, sodium, potassium, ligand-gated channels. You already know concentration of sodium is higher outside of the cell, while concentration of potassium is higher inside. So at this point, sodium diffuses into the cell, potassium diffuses out of the cell. However, turns out, for this particular channel, there's much more sodium diffusing within than potassium going out. So overall balance results in more positive charges getting into the cell, which means local depolarization of that particular part of the sarcolemma. This local depolarization is called endplate potential. It's the graded potential. Remember, graded potential can be of a different value, right? Now, if this graded potential reaches a specific threshold, just like in the nerve transmission, here's what happens. When it reaches the threshold, it opens now sodium channel, which is, since it's the potential that opens it, it's voltage gated. Does that make sense so far? Okay? Good? So, sodium rushes into the cell, and this results in depolarization, basically action potential. Okay, we're good? So if we wanted to draw a graph, it's going to look very much like what you saw for the nervous system. Okay? So you have time and voltage, and you're going to have depolarization here. Okay, this point, when a part of the membrane is completely depolarized, sodium channels are going to close. Just like in the nervous system, they aren't just closing, they're completely blocked. Does that make sense? And nothing can open them. Now, repolarization occurs due to the efflux of potassium. Okay, so we can write it down this way, that depolarization happens because sodium goes in via voltage-gated channels. Repolarization occurs due to the potassium going out via voltage-gated channels as well. Okay? Same story. Slight differences, there's no hyperpolarization. But remember, in the nerve transmission we talked about absolute refractory period. So this absolute refractory period is somewhat, it was, so during this time, nothing can stimulate another action potential. Nothing can create it. Why do we need it? To avoid tetanic contractions. To avoid permanent contracture of the muscle. Does that make sense? There is some period during muscle contraction when nothing can create another action potential. So there's going to be a little bit of a pause. Okay? Good? So we've got this wave. I'm not drawing potassium channel. We've got this wave moving along sarcolemma. Okay? Near have two tubules that, well, formally speaking, increase the surface area of sarcolemma. But not just that. You see what happens? The tubules allow action potential to kind of reach deep into the cell. Does that make sense? So I'm going to draw this action potential wave. Since I don't have any space here, I'm going to move over here. Okay, so it goes here. And, well, it just moves along the membrane, right? Like it's a action potential, for that matter. And it turns out that near each T-tubule, there's so-called, fancy word, terminal cisternae. Okay, so what is that terminal cisternae? This is basically your endoplasmic reticulum or sarcoplasmic reticulum, call it whatever you want. And the function of sarcoplasmic reticulum in the muscle cell is to store calcium. There's plenty of calcium here. So far we good? Now, what I want you to understand conceptually, and part of it is because to save my life I won't remember what are the two sets of receptors that connect sarcolemma to sarcoplasmic reticulum. But my point is this, look, there's action potential running here. Okay? There is a set of receptors that connect sarcolemma to sarcoplasmic reticulum. Am I clear? And when these receptors sense the action potential, they open voltage-gated calcium channels. There's a lot of calcium in the sarcoplasmic reticulum, very little in a sarcoplasm. So calcium diffuses from sarcoplasmic reticulum into the sarcoplasm. Does that make sense? I'm going to walk you through the whole story one more time. So let's do this from the very beginning. Action potential arrives at the presynaptic terminal of the lower motor neuron that results in the release of neurotransmitter, which is acetylcholine, into the synaptic cleft of the neuromuscular junction. Acetylcholine binds to sodium-potassium ligand-gated channel, opens it, and ion sodium diffuses into the muscle cell while potassium diffuses out. There's more sodium diffusing in. That overall positive influx results in the local depolarization, which we call endplate potential. If endplate potential is sufficient, it results in the opening of voltage-gated sodium channels, generating action potential. Action potential, here the details, is propagated via sarcolemma until it reaches T-tubule. Well, not at end, reaches T-tubule. When it reaches a T-tubule, there are adjacent terminal cisternae, forming a so-called terminal triad. Fancy name. But the point is, this action potential is sensed by special receptors here, okay? And that sensing causes the opening of voltage-gated calcium channels on the sarcoplasmic reticulum and release of calcium into the sarcoplasm. It's a multi-step process, but this is another example of a thing that is good for the brain dump. Okay? We good? Is that understood? Awesome. So, this is excitation. Next step that we're going to chat, and I don't want to interrupt because it's kind of smooth flow, is contraction. So calcium is the stimulus for the contraction. Am I clear? Now, conceptually, I want you to understand this. As long as calcium is present in the sarcoplasm in a sufficient amount, there will be contraction. But for calcium, there are two processes that happen at the same time. I'm not going to put this on the board, I don't want to mess up the schematics. But think of it this way. There are channels that permit diffusion, okay? Are you with me? And there are pumps that return calcium back into the sarcoplasmic reticulum. When channels are open, pumps can't compete. You see what I'm saying? The minute all of this stops, the minute ACH is not released anymore, channel is going to close. And pumps will pump calcium back, stopping the contraction. Good? Now, let's talk about the mechanism of contraction, kind of linked to the sarcomere story. So I mentioned that there are thick and thin filaments, right? So this is your thick filament. Okay. I'm going to call it myosin filament. It's a thick filament with a head. This is your thin filament, the actin. Well, it turns out that when there's no calcium, here's what happens. Actin is surrounded by the protein called tropomyosin. Okay? And this tropomyosin does not let actin and myosin to interact. It blocks actin from binding to myosin. We good so far? Well, on top of that, there's another protein here bound to tropomyosin called troponin. Right? So when calcium gets into the mix, this is what happens. Look. Calcium binds to troponin, and this binding induces the change in the shape of tropomyosin, which liberates actin, and actin now can form a cross bridge with the myosin. That makes sense? A student of mine previously taught me a really fun and extremely corny, analogy here. So myosin here is the boyfriend, and actin is the girlfriend. Okay? Now, why a girlfriend cannot date a boyfriend? Because her dad doesn't let her. Okay? But who rules the dad? Mom. So when they have a friend visiting, friend talks to mom, mom pulls dad into the conversation, dad loses control, and now girlfriend and the boyfriend can finally go on the date. That makes sense? But that's actually a pretty good way to remember that. I actually saw students just writing this down before the exam as a brain dump. Okay? Again, as long as calcium is present, this is going to happen. Does that make sense? Good? Now, one more thing that we're going to do. I'm not going to draw it because I can't, really. I'm not capable of doing that. We're going to illustrate the power stroke. So once the link is established, right? So what's going to happen next? Well, you remember that the head of the myosin can not only bind to actin, it can also break down ATP. Does that make sense? So in the following illustration, my hand will be the head of the myosin protein. Does that make sense? My arm will be kind of the rest of it. Okay? Keys is the ATP. Okay? The ruler is actin. So myosin head, check this out. Myosin head breaks down ATP. It's broken down. See that? When ATP is broken down, what is released? Energy. This energy is used to cock back the myosin head, like you cock back the trigger on a handgun. Does that make sense? The head binds to actin. See that? And releases the ADP and phosphate. When ADP and phosphate are released, the head of the myosin propels actin towards stem line. So far, so good? Now, here's the problem. It's stuck. It's really hard to operate it with one hand. You know what? These are going to be ATP molecules, right? So it's stuck. Okay? What should happen next to release the head from the actin? It's got to bind ATP. So bound ATP breaks it down. Breakdown releases energy, charges back. Binds to actin again. Releases ADP and phosphate, propels it back. What should happen next? It's got to bind another molecule of ATP to get released. Does that make sense? Breaks it down, charges, binds, releases, propels it forward.